Good evening to all of you. I am Dr. Anushka Rajpaksha from University of Sri Jayawardhanapura, moderator of this event. I welcome all of you to webinar on microplastic in the ecosphere, extractions and identification techniques. This workshop is jointly organized by Young Scientists Forum, initiated by National Science and Technology Commission and Ecosphere Resilience Research Center, University of Sri Jayawardhanapura. Microplastic contamination in ecosphere is now a global issue. However, knowledge is limited for identification techniques and their analysis methodologies. Therefore, this webinar is organized to facilitate and give participants an overview of the overall microplastic contamination, analysis, and extraction techniques. It's a great pleasure and honor to introduce today's resource person, Professor Mettika Vithanage. Professor Mettika Vithanage is a professor in natural resources and the founding director at the Ecosphere Resilience Research Center, University of Sri Jayawardhanapura. She is now serving as a professor in the Institute of Agriculture, University of Western Australia, and National Institute of Fundamental Studies, Sri Lanka. Her academic background covers water chemistry, environmental remediation of toxic metals, antibiotics, agrochemicals, microplastic, and waste biomass conversion. She was selected as a fellow of the National Academy of Sciences in Sri Lanka in 2022. She is a highly cited researcher 2021 by Clarivate and the prestigious Faisal Al-Karafi Award from the World Academy of Sciences in 2020. She was awarded as Most Outstanding Senior Researcher in Physical Sciences by the Committee of Vice Chancellors and Directors, Sri Lanka, in the year 2020. She has received many awards, including Presidential Awards for Scientific Publications for 12 years from the National Research Council of Sri Lanka. She has secured more than $500,000 US from national and international grants. She became the chairperson of this forum, Young Scientists Forum, in 2017 and awarded as the best young scientist in the year 2018 by the Young Scientist Forum of the National Science and Technology Commission, Sri Lanka. Further, she was listed as top 2% of the most cited scientists across various disciplines globally in the year 2017, 2019, 2020, and 2021. She has contributed over 225 SCI Index journal articles, over 50 book chapters, and 10 co-edited books. Her citation record is over 19,000 with an H index of 66. Now, I cordially invite Professor Mettika to begin this workshop. Over to you, Professor Mettika. Thank you.
Thank you very much, Dr. Anushka Rajpaksha. Can you make the screen sharing possible? Thank you very much for the kind invitation from Young Scientist Forum, NASDAQ, to give this, to deliver this webinar on microplastics, especially to understand the extraction techniques and how to identify the microplastics in environmental samples. So let's start it. So microplastics are now considered as an emerging pollutant in the environment in almost all environmental matrices, especially considering the marine ecosystem, aquatic ecosystem, and soil, as well as in the atmosphere. So when you have microplastics in all three spheres, all three physical spheres, hydrosphere, lithosphere, and the atmosphere, that means soil, water, and air, it is automatically coming into the biological systems as well. So especially in the human and animals can also be having microplastics in their system. So then all four spheres, biosphere, plus three physical spheres are definitely contaminated with microplastics as right now. So there is a classification of plastic litter based on the size of plastic that we see in the environment. Our focus is mainly on microplastics. So you can see meso, macro, and mega plastics are more than 5 millimeter in size, and nanoplastics are less than 1 micrometer in size. So this is how the classification of plastic litter is done in the environmental samples. So what exactly are microplastics? As we saw in the last slide, we know the microplastics are the plastics that we find in the environment, which is in the range of 1 micrometer to 5 millimeters. So it's a vast range. There are two different types of microplastics in the environment, primary microplastics and secondary microplastics. So primary microplastics are the microplastics that are being used for manufacturing of different other plastic products. So there are plastic resin pellets. As an example, you must remember the Express Pearl incident. I'm sure all of you have seen the plastic pellets that was discharged into our beaches. And those plastic pellets, we call it as plastic resin pellets. So those are used as a raw material for plastic production. Other than that, there are microbeads. So these microbeads are very small powder-like plastic particles, rounded particles. These particles are being used to produce personal care products and cosmetics and so on. Especially like toothpaste and other personal care products that you apply for scrubbing and so on. So these are consist of microbeads. So there are various different types of microbeads, and they are being used in paints, in other coatings, especially for textiles, and so on. So there are primary microplastics that we use to produce the other plastic materials that we use for our general purposes. These are the primary microplastics. So it is very hard to see primary microplastics in the environmental matrices because of their smaller size. It is, you can't say it is not present, it is present. But since they are very, very small, it is very hard for you to see in the naked eye. But the secondary microplastics, you see everywhere in the environment. Wherever you go, you will see the secondary microplastics. Secondary microplastics are the plastic, smaller plastic particles that are being produced by the degradation of the big plastics that we are throwing out to the environment. So they mainly by mechanical breaking or by the UV radiation, biological activities, there can be various different causes to form secondary microplastics from the primary microplastics. So mainly, like the UV radiation from the sunlight is the main agent which causes the bigger plastic particles to break into smaller microplastics. Especially, the plastics are being made up of these polymers, polymer chains. So polymer chains can be breakdown from the UV radiation. Then when you have the weaker polymer chain, it can be broken by the mechanical forces, biological agents, and so on. So that you will have smaller pieces of plastics forming in the environment. So mainly the photooxidation, then starts hydrolysis and biodegradation. So these are the three major processes for formation of secondary microplastics in the environment. So we really need to understand how the microplastics are being formed in the environment, so that if there are causes, like agents, we can stop or reduce, then we can try to make that happen. But it is it is not very possible because of the they are also from the natural sunshine and all these biological activities and so on. So there are various different types of microplastics in the environment with various different shapes, right? So when it comes to types of microplastics, it is mainly the polymer type. So can be polyethylene, can be polypropylene, PVC, polystyrene, and thousands of different types of polymers are present in the environment as types of microplastics. Then you see in this picture, you can see different shapes of microplastics also in the environment. There are fragments, pellets, fibers, foams, and films. So these are the 5 major different plastic shapes that you can see in the environment. So I'm giving an overall introduction to this, different types of plastics, how plastics are being formed in the environment, and what are the major, primary microplastics and the secondary microplastics are the major microplastics that you see in the environment and their sources. So we have discussed in brief all these, the general background regarding the microplastics. So we are entering now into the main theme of the webinar today. How are we going to extract and identify the microplastics in the environment? So before you go for extraction, you do sampling, right? You have to do sampling of various different types of samples that you intend to do in the environment for assessing microplastics. So there are different types of samples that we check for microplastics presence. Biological samples, obviously, fish, and mollusks and water, these like biological organisms in water. So they are the biological samples that we can consider as samples for microplastics. Other than these marine organisms and organisms in water, there are organisms in the soil, like nematodes, like worms, and then maybe like lichens and mosses in the on the trees. So they are also biological samples where we can analyze them for microplastics as well. Then it comes to the waste waters. So when you call it wastewaters, there can be landfill leachates coming from various different landfills like municipal solid waste dump sites and so on, or industrial wastewaters and domestic wastewaters. Probably like wastewater treatment plants. So there are different types of wastewaters that we sample for microplastic analysis. Then the drinking water. When you call it as drinking water, there can be drinking water from, like rivers. As an example, River Kalu, River Kalani in Sri Lanka, we use it for drinking water purposes, like any other rivers, like even Mahavali. So those river waters can be taken as samples for microplastic analysis or sampling. And seawater, of course, we are using seawater to generate salt. And then water in PET bottles, right? The drinking water bottles. So those are also be sampled as water samples for microplastics. Then sediment and soil samples, right? Sediment and soil samples, especially we when we talk about microplastics, we think automatically we think about the marine ecosystem and waters. But you must know the majority of microplastics are found in soil in the globe, right? You may wonder why, how come soil has microplastics? Soil has microplastics especially from plastic mulch films. So, not very much popular in Sri Lanka, but most of the temperate countries, they use plastic mulch films to protect the soil from evaporation and erosion and so on. So these plastic mulch films, when it exposed to the sunlight, starts degrading. So after one season, you can't remove the plastic mulch film, so it will automatically decompose or degraded by ploughing and by the UV radiation and so on. So that is one such method of plastic mulch, microplastic to be found in soil. Other than that, you apply various different fertilizers, thinking that organic fertilizers, so-called organic fertilizers, like compost, municipal solid waste compost. There are a whole lot of microplastics in municipal solid waste compost. And then you apply wastewater treatment sludge as a fertilizer to soil, agricultural soil. So this wastewater treatment sludge definitely have microplastics in it from the shampoo and toothpaste and whatever we use microplastic beads consisting stuff in our day-to-day life. So those are the different types of samples that we use for microplastic sampling. Other than that, you use atmospheric, like air. You suck air, or you keep some bottle or something to get the dust deposition from air. So those also can contain, can be as samples for microplastic sampling. When you have a sample for microplastic analysis or extraction, there is a pre-preparation. So pre-preparation method can be different depending on the type of sample that you use. Let's say if you're using biological samples to analyze for microplastics, then you have to dissect those biological samples in the proper manner, right? Especially, you need to find the microplastics in the gastrointestinal tract or wherever the sac is, what is eating goes in. So these places you have to dissect very carefully. If it is like wastewater samples, you can directly sieve the wastewater into various sieve sets, sieve stacking, and then you can collect the samples. That is kind of a pre-separation. If it is like sediment samples, then you collect the sediment core or maybe the sample that you use for sampling. Then you are using separation methods. There are various different separation methods. The most common method for microplastic sampling, microplastic extraction is the density separation method. Other than that, you can use digestion as well and filtration. So by density separation, digestion, like separated, density separated sample can be digested and then filtered to take the plastics. Then you are using various different analytical techniques to analyze the microplastics in your sample. So it can be the visual analysis. If it is visible enough, you can use visual analysis like naked eye. Or if it is smaller than, smaller, then you can go for a microscope, even stereo microscope, optical microscope. If it is much more smaller, you can go for like SEM, scanning electron microscopy and so on. But these analytical methods will not give you an identification of the type of the microplastic, like what type of polymer is present. So for that, you are using vibrational spectroscopy, especially Raman and FTIR. So those are the two most common techniques to understand, to identify the type of microplastics in your sample. If you need to go further, like for nanoplastics and for like, to identify the peaks and so on, to have more sensitivity analysis, then you can go for mass spectroscopic analysis like GCMS. So this is the structure of sampling, separation, digestion, and analysis pathway for microplastics in the environment. So as I told you, there are different types of sampling methods. So when you do research for microplastics, you really need to go for standard sampling methods. Otherwise, it will be very hard for you to validate your data. So there are standard sampling protocols for microplastic sampling in water specifically, right? So this Newton-Tow method is the most standard method. So there is a net. That particular net has particular width, rectangular type of mouth, and then you have a bigger net in the to drag in water and collect whatever the samples water, especially in surface waters. Other than that, you can use grab sampling. In the case of grab sampling, you can just drop this net in one particular place and grab the sample up. So you can drag it and you can grab it. So these are the two different sampling techniques used in microplastic sampling. If you don't have these kind of sophisticated nets and sampling equipments, you can use a metal bucket and get a sample from freshwater body. The most important thing that you should remember is you have to minimize use of plastic stuff when you do sampling and, digestion and all this, the protocol that we talked about in the last slide. So, it is very hard for us to avoid plastics, because plastics have been like very common and present in almost everywhere, every thing that we use. So it is unavoidable actually in the real meaning, but you have to avoid plastics as much as possible when you do sampling. So the best thing is either you use glass bottles, or either you use metal samplers for plastic sampling. So there are different types of nets, as I mentioned earlier. Manta net, Newton net. So these nets are being made of aluminium or some kind of, metals that are very light. Some people are using plankton nets for microplastic sampling, but it is not very standard because plankton net is already made up of microplastics, like plastics or polymers. So therefore, it is not really encouraging for people to use plankton nets for plastic sampling. It is being used in many research papers, but there is a possibility for questioning by reviewers regarding the control sample of the sampling when you use a plankton net for microplastic sampling. So after you take the sample, you have to carefully collect the materials present in the net, and you have to put them into a sequenced sieve set, right? By putting it into a sequenced sieve set, you can pour water, and then the sieving, different sieve sizes will be extracted, can be taken. So this is the most common method of microplastic sampling in water. So it is very easy and straightforward to do microplastic sampling in water, right? So when you put the sample into the sieve set, then you can pour water, and then you can rinse the debris slowly, and then take each sieve and separate them after drying. Once you have the sample in these sieves, you have to do the extraction. So how do you do the extraction is different. So normally there are, various people are using different types of techniques for separation or extraction of microplastics from the sieved sample. Either coagulation or flocculation. You can put, different types of chemicals and make it as a flock, or like alum and lime, you can put and make it a coagulant and deposit. Filtration you can do. These are not very applicable in the laboratories. These are, like used in bigger scale. Sedimentation and magnetic treatment. So these are the different types of techniques that are being used in larger scale to extract or remove microplastics from water. But in the laboratory, you can just use a salt water, a saturated salt water solution, and then pour that salt water solution to whatever the sample that you collected, and then the microplastics will be floating in the on the top. So floating stuff, whatever the stuff that is being floating, will not be only microplastic. There can be like plant materials and so on. So then you have to do wet peroxide digestion to remove these organic, like natural organic matter, like plant materials, plant debris, and so on. So we use wet peroxide digestion commonly, like 30% hydrogen peroxide is used with 0.505 molar ferrous solution. So that is called Fenton reagent. And if you heat this particular sample to 70 degrees Celsius, then all these organic matter, especially the plant debris, will be dissolved. And then the remove whatever remaining is the plastic material. So then you can filter and use it. So after the digestion, you can maybe use different types of filter papers, maybe, and filter the material, filter the solution or whatever the supernatant, and take the supernatant filtered stuff and dry it, carefully dry it. Why I say carefully dry it? Because when you have microplastics are very light. So a small amount of air can take away all your microplastics in the sample. So you need to dry it very carefully without this, getting your sample flow away. So this is the most common extraction techniques that we use in our laboratories. We use various different types of salt solutions. Sorry that sodium chloride, the chloride C is simple here. I'm very sorry about it. It should be NaC capital and L. So sodium chloride is the most common salt solution that we use for plastic extraction, because it is very cost effective, right? Very cheap. So, you don't need to get the sodium chloride from laboratory supplies, but you can use the table salt. Only thing is you need to make a saturated salt solution. You all know how to do a saturated salt solution. Other than sodium chloride, there can be various different other, like salt solutions like potassium chloride, magnesium chloride, zinc chloride. Zinc chloride is the best, but it is a bit toxic. Sodium iodide is also very, very good salt solution to extract microplastics, but it is also toxic. Sodium bromide also heavily used, but these are toxic and highly costly for people like scientists in our country. So we use sodium chloride in general. Why do you have various different types of salt solutions? Is because depending on the density of the salt solution, the extraction efficiency of microplastics can be varied. So let's say if you have PVC kind of microplastics in your sample, sodium chloride is not very efficient, right? Because the density of sodium chloride is not great as sodium iodide or zinc chloride. So if you have heavy microplastics like PVC, then you must use zinc chloride or sodium iodide kind of a salt solution, a denser salt solution. So when you select your extractant, you must have an idea about what kind of plastics that can be in your sample. So let's say if you have a sample from landfill, you have a leachate sample, or you have a sediment sample from a leachate draining canal from a municipal solid waste dump site. Then there can be various different types of plastics. So definitely you must use sodium zinc chloride or sodium iodide in that case. If you do sample, if you extract a sample of let's say agricultural soil, then most of the time the microplastics will be polyethylene, polypropylene kind of, so that you can use sodium chloride in that kind of a sample, for that kind of a sample. So you must read and have an prior understanding about your sample and also the density of your soil solution. So based on the density of your soil solution, you can think of whether this particular soil solution will be efficient enough for extracting microplastics in my sample. So you have sample, and then you have some sieved portion. Then that sieved portion is, you take a known amount, known weight, let's say 10 grams of soil, microplastic contaminated soil. You take 10 grams of soil, and then you put 100 milliliter of saturated soil solution. Then you stir it vigorously for like 5-10 minutes, and then you keep it for settling. Once it is settled, or the soil has been gone down, settle on the bottom, then slowly, carefully you pour the supernatant through a filter paper, right? If you think that your your sample has quite a lot of organic matter, before filtration, you must put the Fenton reagent, H2O2 and ferrous sulfate solution, so that you digest, you then heat it up for 75 degrees Celsius for like half an hour, and then you digest it, and then you can filter it. So depending on the sample, if you are using a soil solution, soil sample, then definitely you have a lot of organic material from plant residue and so on. So that you definitely need the Fenton reagent to digest. If you use water samples, then this digestion is not very important. You can just filter it through a sieve stack and then use it for identification. So it depends on what type of sample that you deal with. If you are using compost sample means that everything is lighter, plastics plus the organic material from, like various organic residue, so that you do oil extraction, right? You can do oil extraction. You can put a little bit of oil on top. So all the microplastics will be attached to this oil, and then you can do the extraction again. So what I'm telling here is, depending on the type of the sample, that you have to do various, modifications for your extraction. Extract and solution you have to choose depending on that, depending on the sample, and digestion, whether you have to select digestion depending on the sample, and so on. So these things you need to read and understand. By reading many papers, you can understand how do you select the best extraction methodology for your sample, right? So this is again another example to show various different types of microplastic extractants, like with sodium chloride. So then the most important thing is once you extract the sample, you must repeat it for at least 3 times, because there can be smaller microplastic particles still remaining attached to soil, attached to organic matter, and so on in the same sample. So you have to repeat the same procedure 2 more times, so that you can be clear enough that almost all the microplastics must have come to your supernatant. Okay, so repetition is important. For that repetition, you can reuse the same salt solution. There is no issue, right? Because salt solution is just for the density separation. So because of the density, the plastic separates. So the same salt solution, you can use it for many times, right? Many times you can use it. So extraction processes, if you have like solid food, let's say like you have a fish, then the extraction procedure will be different, right? Even if it is like liquid foods, like let's say you have beverage, fruit juice. So then the extraction protocol will be different from what we talked, before. So solid foods and liquid foods, you have to do the chemical digestion first, right? First, you do the chemical digestion, because there are various different, like flesh and so on. So you have to do the chemical digestion and then go for the plastic extraction by density separation or microfiltration, right? You can, you don't need to do density separation, because if you can clearly see, after the solid and liquids are being digested, there will be only a solution. So that solution you can filter through and then take the sample for, microplastic analysis. So when it comes to atmospheric sampling, the sampling protocol is a little bit different. There can be like passive sampling and active sampling. In the case of passive sampling, you can, use kind of a vacuum cleaner or vacuum or sucking machine to do the passive sampling, right? It is very cheaper and easy. Then you can take the whatever the accumulated in your say the machine that you use, and then do the density separation. So active samplers means there is a motor, and from the motor, vacuum is being created, and you suck the air inside a chamber. So that particular chamber gets all the particles accumulated. So that is called as active sampling, where you use for, like dust sampling, especially the particulate matter sampling. That can also be used for sampling air for microplastics. But this is a little bit expensive. Passive sampling is the most cheapest method for air sampling of microplastics, and it can be easily used. You can even, like keep 2 bottles, or you can, keep a wide mouth bottle somewhere, and then like after 7 days, after 5 days, you can take that bottle and see. Then there might be, dust collected inside the bottle. That sample can be used for salt extraction, saturated salt solution, and extract samples for microplastics. So that is also possible. Like any other sampling protocol, there are challenges in atmospheric microplastic sampling. Not only atmospheric microplastic, there can be challenges for, like water sampling, soil sampling as well. Especially when it comes to, avoiding plastic material during sampling is another type of a challenge for almost all, sampling like soil and water. So you need to use at least, like either a metal containers or either glass containers without using any type of plastic containers. In the case of atmospheric plastic microplastic sampling, you have to make different devices, like particulate fallout collectors, like ambient filter samplers, kind of, different types of samplers you need to make, because there should be a standard way to consider like what kind of volume of air that you have drawn to collect the particular microplastic amount. Because in water, when you sample from a river, how many liters that you have poured for your filtration device or whatever the sieve set. So normally, if it is river water, and if we use by sampling, not dragging, but deploying the sampler, or deploying a bucket, metal bucket, and then we take the volume of the bucket, and then we calculate the amount of water that we pour through the sieve set. So we know whether it is 10 liters. So the sample that we have is, let's say we have 5 microplastics coming from these 10 liters. So we know per liter how many. So we always quantify, right? When you present it in the scientific audience. So in that case, you need to always calculate the amount of sample that you are taking. If it is soil sample, 1 kilo of soil, right? If you take 1 kilo of soil or 100 gram of soil, then you can calculate the microplastic amount per kilogram, right? Per liter. In the case of atmospheric sampling, that is another challenge, so that you need to use particular way or per day or per volume of, like meter cube of air or something like that. And extraction techniques are very common, we all know. So the aerosols, whatever the sample, atmospheric dust sample, we do the digestion and density separation, then filtration and use it for analysis. So this is the challenge that I was talking about. We have the challenge from the background contamination, because whatever we are using, there can be a contamination of plastics because we are using quite a lot of plastic materials, even the clothes like lab coat. If the lab coat has some, like fibers of plastic, nylon, then can be contaminated. So you have to be very careful. You have to run a control sample, few control samples as much as possible. Otherwise, there can be easy contamination from the background. So that is very important aspect to be remembered, to keep in mind, especially when you do microplastic analysis in the laboratories, because there are thousands of synthetic polymers that we use every, like even pen, pen, whatever the specs, or all these things have plastics. So if there are degradations and, contaminations, there can be easy contamination and your sample can be easily contaminated. And there are challenges of the digestion method that you are using, because there can be, like some organic materials may not be digested, and sometimes the microplastic itself can be digested from the digestion liquid that you are using. So I have to be very careful when you select the digestion reagent. So there are various different types of digestion techniques: oxidative digestion, acid digestion, alkali digestion, and enzymatic digestion. So either of these techniques that you can use, but you need to understand whether this particular digestion technique is suitable for your microplastic sample, right? So most common method is the oxidative digestion by like Fenton reagent or H2O2 with iron salt. You can use acid digestion as well, but acid digestion, by doing the acid digestion, the sample color can be changed. And alkali digestion, there can be destruction of the nylons and some other polymer types. Enzymatic digestion is also possible, but it is not used quite frequently. So depending on the sample that you have, you need to consider the digestion technique as well. So still there are no standard protocols for microplastic analysis. Like, let's say when you take a compost sample, this particular extractant salt solution to be used, this particular digestion technique to be used, there is no standard protocol. So depending on the protocol that the scientist is using, the results can be affected. So that you have to keep in mind, especially when you compare like results from one study to another. So you need to think about all these things. So challenges with the density separation is being described in this slide nicely. So if you have like water, then there will be only very less number of microplastics will be floating, right? If you use sodium chloride, the density is little higher than water, so there can be more microplastics floating. But if you use like a salt solution kind of sodium bromide, which has very high density, like 1.53, so there can be a lot of different types of microplastics can be floating. So that is also, again, another time I'm mentioning, depending on the sample that you are trying to analyze, think what kind of salt solution that you will be using, right? Especially like if you're using sea sediment or like beach sediment sample, there can be various different types of polymers. So better you use like sodium bromide or zinc chloride or, like much more denser solute. Once you have the extracted sample, then it is quite easy, quite standard, quite straightforward regarding the identification, right? So when like the sampling, extraction, digestion, these are the manual methods, like you had to put a lot of efforts for those things. So it is heavily, human hours are being taken for these microplastic sampling, extraction, and digestion. But when you have it sieved and collected as a sample, identification is very, very straightforward and easy, right? So as I mentioned to you earlier in the beginning, so easily we can use FTIR, which is a very easy technique that anybody can use. And FTIR is mostly available in, like commonly available in material science laboratories. And if not, you can use just a microscope or naked eye analysis for identification techniques. So chemical characterization of microplastics that you analyze or you sample is very important. So for chemical characterization, we use either FTIR, Fourier transform infrared spectroscopy, or Raman spectroscopy. Since these techniques are already coming up with libraries, so polymer libraries are inbuilt in these instruments, so that when you put a sample, it automatically gives the polymer type. So there is no big effort that we need to use or we need to put to identify the sample. So that is why I told you it is very straightforward. If you have an FTIR, you can easily identify the polymer type of the sample of the microplastic that you have. So FTIR is the most common method. Raman is not available in many laboratories, but FTIR is available in most materials and laboratories. So you can easily use FTIR to analyze microplastics. So depending on the size of the plastic particle, there can be various different types of FTIRs for you to use. But we know that microplastics means the range is much larger. So it is like 1 millimeter, 5 millimeter, then you can clearly see. So you can use the ATR mode of FTIR, the most common FTIR, to analyze microplastic type. Right? So this particular slide give you a good idea about different types of FTIR that you have. And now, because microplastics is becoming an emerging contaminant in the environment, FTIR techniques are being developing, technologically advancing to analyze even nanoplastics in the environmental samples. So there are, even microscope based FTIR. So if you have a microscope imaging FTIR, then you can easily detect very small, tiny, like microplastic particles, let's say the plastic particles in your water bottles, PET bottles. You can easily identify using microscope-based FTIR. There you can either use a dye to stain it and then use the microscope to analyze. It's very easy. And if not, you can go for Raman technique. So there are different types of FTIRs available even in Sri Lanka, which can be used for various different types of microplastic analysis. So that's all for regarding, microplastic identification, extraction, identification, and analysis. We have, like in the Ecosphere Resilience Research Center has published like more than 20 papers regarding microplastics. So if you need to go look at the methods that we use to analyze various different like beach sediments, food items, and water, soil, compost, landfill leachate, and many more. So you can go download our papers, or you can send me an email so that we can send the papers and you can read and learn different types of techniques that you can use to extract, sample, digest, and identify microplastics in various different matrices from food, from soil to atmospheric samples to landfill leachate to beach sediments and marine sediment and water and so on. So these are some of the examples of our studies on microplastics from the Ecosphere Resilience Research Center of University of Sri Jawadanapura. There are various other researchers in University of Peradeniya, University of Open University of Sri Lanka, and different research institutions do research on microplastics right now, even Ruhuna University of Sri Lanka. Sabaragama University of Sri Lanka. So there are various different universities now coming into do a lot of research on microplastics. So we can support, we can do analytical support, we can advise you to how to do sampling, how to select the best extractant and so on. With regard to your research questions. So that's all. I'm very thankful to Dr. Anushka Rajapaksha, Director, Ecosphere Resilience Research Center, University of Sri Jawadanapura, and as a member of the Young Scientist Forum of NASDAQ for organizing this webinar. And I'm very thankful to the Young Scientist Forum to giving me, inviting me to deliver this webinar to this eminent audience. If you have any questions, we can discuss about those questions. I can answer whatever the questions that you have regarding microplastics. Thank you very much.
Thank you, Professor Mehtika. The
Professor
session is
Mehtika, I'm
open
Kalpa
for
here.
the discussion.
Good evening. Thank you. Good evening, Professor Mehtika. I'm Kalpi here.
Yeah.
Just I want to thank you because you've done a nice presentation and comprehensive lecture. Thank for all on behalf of the YSF. And one simple question I want to ask, because since as long as we are using plastics, we know that will come to the environment, right? That made of microplastic, that's all we know. So then why we go back to the analysis? But the final outcome studying of microplastics, because we know that come from the plastic and we made use from the plastics. Yeah. That's one question. And another one is, now when the polymer is made from the plastics, is there a potential to break down the plastic, or the primary plastics to be break down in the microplastic? Because what I asked this question is, like, the Pearl Harbor incident, the lots of primary plastics were dumped into the sea. Will it cause any microplastics degradation and will be damage to the ocean pollution? Thank you, Mehtika. Yes.
Oh, thank you. Thank you very much, Dr. Kalpa. And answering to your first question, yes, we are now analyzing environmental samples for microplastics is because of the emerging, being an emerging pollutant in the environment. So we actually don't know yet what exactly the health implications of these microplastics. But microplastics have been found in human blood, human placenta, even breast milk, and all these biological samples. So that means there is a potential of contaminating ourselves by microplastics. But we, since it is being really new contaminant or pollutant, we do not know the health implications yet. But there can be huge health implications because it is being a polymer, it is being with various different pathogenic organisms. It might be like microplastics can absorb various different pollutants like antibiotics. So in the environment, so there can be antibiotic resistant organisms present on the surface of microplastics. There are papers already published. So there are potential there is a potential for microplastics being a threat to the human health. So that is why we are, looking at various different environmental samples for microplastics. Moving into your second question, Dr. Kalpa, yes, we all know that there have been a huge issue of plastic pollution because of the Express Pearl ship disaster. So this ship disaster is being considered as the world's largest polymer related disaster from the marine environment. So these are the plastic pellets that we are looking at. These plastic pellets are itself microplastics. So these microplastics are now, we see them in the beaches, and the surface of these plastic pellets have been changed quite rapidly, and the size of the plastic pellets have been reduced very significantly. Now the plastic pellets are now almost half of the size of the pellets that we saw in the May 2021. So there is a huge issue of contaminating our seagrass beds, coral reefs, our fish population from these microplastics from Express Pearl ship. There are two types of microplastics coming from Express Pearl ship. One is the plastic pellet, and the other is the pyrolyzed plastic or burnt plastics. So both can be very dangerous, and our biological system in the marine and coastal environment can be definitely contaminated. Thank you.
Thank you, Professor Mehtika. There's another question in the chat window.
Okay, the very first question in the chat box is from Maheshan. If we use acid digestion to isolate airborne microplastics from biogenic samples, lichens, is there any possibility to dissolve microplastics? There can be a possibility to dissolve microplastics, but that dissolution will be very minimal. But the problem will be the discoloration of your plastics. So when we do microplastic sampling and identification, we categorize plastics based on the color as well, because based on the color we can conclude regarding the source of microplastics, the source and the where it is coming from. So that discoloration might affect your sample. And then there's another question from Isuru. When we consider tiny there can be very small quantity of microplastics in water. Then how we quantify the concentration of microplastics in water? Is there any standard quantification method? Yes. If you are using drinking water for microplastic sampling, you can easily use Nile red staining for analyzing your microplastics. If you can use the Nile red staining and use FTIR microscope, then you can quantify the number of microplastic particles in your water. There are many papers you can just read, and see thousands of papers now regarding microplastics in the environmental matrices. Another question from Ureshi, if we are extracting microplastics from biological samples such as seafood and fish, do we have to specially consider about the sample storage temperature until the extraction part? Most of the, it depends, especially this -20, -80, -40 degrees Celsius is because of the biological sample to be preserved. If not, biological sample, the fish, flesh will be degrading, decomposing, right? Deteriorating. So that might affect the microplastic. So it might, like dissolve with the smaller particle and, flow it and so on. So to keep the biological sample to be preserved, we are using minus 20, 80 degrees Celsius. It is not to preserve the microplastic, but to preserve the matrix of the microplastic in the sample, right? There is no harm for the microplastic from that. Another question from Amasha, collecting organic sample from aquatic environment. The same question, I think I answered that. Gayan is asking how to minimize background pollution in post-mortem human tissues in order to detect the presence of microplastic or quantify them. Can you provide assistance? Yeah, there is a researcher in the University of Sri Jagadhanpur in the medical faculty. He is using microplastics in human tissues and stem cells and so on. So you can contact him, or either there are a lot of papers available regarding microplastic to minimize the background pollution on post-mortem as well. So the best thing is using glass ware or metal ware to reduce the background pollution. Otherwise, it will be pretty hard. So that is what I can say about that. I'm not very familiar with the human tissues, but we can find some papers. If you are interested in, you can send an email. So I can try to find some papers for you. Isuru again is asking another question. Wet sieving for separating microplastics. Yes, wet sieving is, it is the method that I told you when you sample water from rivers and so on, you can use wet sieving. Even for soil sample, you can use wet sieving. Wet sieving means you have stack of sieves. Let's say you have three types or three different sieves, 1 millimeter sieve, and then 0.5 millimeter, and then 0.25 millimeter line. So you have stack of sieves, and you pour or you put the soil sample and you pour water. So with the water, the sample, goes down and it sieves down. So it is very common, and you can easily use in Sri Lanka. We do it in our lab. We have already bought some stainless steel measures in different sizes, and we prepared our own sieve stack, and we do wet sieving. We can even show it to you if you want to visit our lab, or we can send some photos or videos for you to learn. So that's all in the chat box. If there's any other questions, I can answer. Another question came up. Is it harmful to use zinc chloride? Zinc chloride is not that harmful, but if you are using sodium bromide and sodium iodide, it is toxic. Zinc chloride is harmful in terms of the environment, because after you use the zinc chloride, then you throw it to the environment. So zinc will be adding into the environment. So you have to be very careful. It is not that environmentally friendly, but sodium chloride is fine. But if you really need to use zinc chloride, that is not a big deal. But sodium bromide and sodium iodide, you have to be very careful if you're using. Thank you. Any other questions? If not, we can wind up, right?
Yes, Professor Mithika. On behalf of Young Scientists Forum, I would like to give our sincere appreciation to you, Professor Mithika, for this very informative and interesting session. Also, I would like to convey our sincere gratitude to Director, National Science and Technology Commission for giving approval and the arrangement for this session. Also, I would like to thank Chairperson of YSF 2023 year, Dr. Kalpa Samarakon, and all steering committee members, and special appreciation to Ms. Tilini from National Science and Technology Commission for her assistance. Thank you all for your participation and for being with us. Thank you.